We have tiny blood vessels in the brain. So if you don't exercise, if you don't make sure that your heart is pumping at a high rate regularly during the week, those tiny vessels that are even smaller than your hair in terms of diameter, they will start to collapse. And you're going to have, and remember the capillaries, it represents 90% of your brain vasculature, all these tiny blood vessels. So if you don't do enough exercise, you will start chronically to have some vessels that will basically constrict and collapse and disappear. Meaning that the surrounding neurons that are here, and they need oxygen and nutrients and everything from these vessels. If these vessels disappear, you're going to lose neurons, right? So exercise is the number one thing. You have to do that, not only for the brain, obviously, for the heart, it's true, for the eyes, obviously. If you don't exercise, you're going to tend to lose eye vision quicker than a normal person as you age. But very important for the brain. You're gonna turn, if you're prone to go to cognitive decline because, let's say, you have a major genetic risk, but if on top of that you don't exercise, you're gonna accelerate, as we know from studies. And that's one of the biggest things we have to put that, we have to put to the world, like, okay, you need to exercise that no matter what. I mean, if you want to stay healthy in terms of brain function, no other choice.
Hi, everyone. Today, my guest is Dr. Axel Montagne, who is a professor and group leader at the Dementia Research Institute at the University of Edinburgh in the UK. I'm personally really excited to have Axel on today because we are going to be discussing really a new paradigm for dementia and Alzheimer's disease. Often when we talk about Alzheimer's disease in particular, there's so many ideas floating around. Is it brain diabetes? Is it amyloid-beta plaques? So rather than speaking to the role of amyloid or metabolism, today I'm hoping to discuss a new paradigm of research that suggests not only an important way of understanding dementia and Alzheimer's, but also how to prevent it. So Axel, thank you so much for coming on the show today. Can you talk a little bit about this idea that dementia can be a vascular disease?
Yes. Hi. Thank you for the invitation. First of all, very happy to be here and talking about that. Yes. So I think the word new paradigm, it's correct and not correct at the same time. It's been several years and there are many studies back from 20, 30 years ago that shows that when you look at a post-mortem brain sample on tissue, just the tissue of people that died from Alzheimer's disease, you can see a lot of vascular problems on every single case. And it has been shown like 20, 30 years ago. And but people didn't really pay attention to that vascular phenotype that we were seeing on post-mortem brain. It's just, I would say, probably the last 10 years or so that we are really tackling this because we have more and more evidence that shows that the vascular dysfunction, and we're going to go into details a bit later, but the brain vessels, so the blood vessels, starts to be dysfunctional and leaky and not doing their job to make sure the brain functions normally. And that's something that we can detect very early on. That is something that is happening with normal aging. And we know now that this is something that is accelerated in people that go towards dementia. And this is true in Alzheimer's disease, which is the major cause. I mean, the main dementia form. There's also small vessel disease. We'll talk about this. There's vascular dementia. There's different kinds. But the commonality about all these dementias is they have a vascular component that is quite early in the disease. And we want, me as a lab and different groups in the world, try to understand how vascular dysfunction contribute to dementia. And that's something now that people pay really attention on.
You were a co-author on a paper showing that about 50% of all dementias start with breakdown of the smallest vessels in the brain. And I mean, this seems very important. So let's talk about how a leaky blood-brain barrier could be even more important than beta amyloid plaques or amyloid beta plaques and tau tangles. What causes that breakdown of the blood-brain barrier?
Yes. So the paper you were mentioning, I guess that's the paper we published in 2020 in Nature, where we show, and we had a few other papers prior to that, where we show that just the fact that we age normally, the vessels start to be normally dysfunctional as we age, like the whole body. It's not only the brain, just not only the vessels. We are just aging. And these vessels, we've developed a way to measure how leaky, if I can use that term, which is easy to understand for everyone, how leaky, meaning that things from the blood can leak out to the brain. And we can measure that with magnetic resonance imaging, MRI. So we developed that technique in the US and California at the University of Southern California, where we can really detect small leakiness of the blood-brain barrier. And what we've noticed is when we scan cognitively normal individuals, young and old, just normal aging, we start seeing some brain regions that are leakier than others. So first of all, there's what we call the blood-brain barrier. There's not only one blood-brain barrier, in my opinion, there's several. And we are trying to understand how they have different properties, different depending on where we look in the brain. But we see special brain regions that starts to leak with normal aging. And these regions, it's a bit, that's the region of the hippocampus, that we call medial temporal lobe. So that's this part of the brain, medial temporal lobe, where we have the major, the learning and memory region that is called the hippocampus. That region, somehow, we still don't know why at that time, is leakier than normal in people that are older versus controls, young. And we know that this region is the region that leaks the most early in dementia as well. So we developed that MRI technique where we can really see visually, okay, where do we have these vascular problems? And interestingly, we also, I guess we're going to talk about this, but not only neuroimaging is important, but we also develop new biomarkers to look at in plasma, so in the blood or in cerebrospinal fluid. We can measure new markers of that gives us an idea of how leaky are the brain blood vessels, basically, to make it simple. And so we have this neuroimaging marker, this biofluid marker, and we show on both that we see with normal aging an increase, what we call blood-brain barrier breakdown, so the vessel disruption in the brain. And we know also in this paper that you mentioned is we can predict future cognitive decline. So the people having more leakiness at baseline when we scan them, or when we measure on their plasma or CSF, cerebrospinal fluid, the level of vascular problems in the brain, we know that they are more, they're going to decline more faster than the people that have low levels of these biomarkers. So we have some predictive value of looking into vascular dysfunction. So to try to link amyloid and tau, which are the classical Alzheimer's hallmark, what we found is the leaky areas that we see in the brain have nothing to do with the areas where we start seeing the buildup of amyloid plaques and tau tangles. So it seems to be two independent pathways, I would say. So you have people that have vascular dysfunction, and in some brain regions, they're going to start having some amyloid plaques, and it doesn't seem to relate to each other. So that's the first point I wanted to make. But also what is interesting to know is when people start to have amyloid plaques, they tend to have what we call cerebral amyloid angiopathy, CAA, which also will disrupt vessels. So you have two schools in research right now. You have people that think that amyloid is something that happens very quickly, very early, that will disrupt the vessels. And you have the other school that I'm part of, that we say that we can see vascular problems without amyloid happening at that stage. And we know that having vascular dysfunction will promote also the formation of amyloid plaques in the brain. So it's still unclear how this is going. What we know for sure is there's early vascular problems, and we need to fix that to make sure that the brain doesn't develop plaques and function normally, and to avoid dementia, possibly.
You mentioned some early diagnostics. I mean, both more sensitive types of MRI to detect some of these, leaky vessels, but also a plasma biomarker. And these were in cognitively normal individuals. I'm not sure what age range, but that seems like extremely important because if you're talking about a new potential diagnostic well before cognitive decline is occurring, it seems like intervention. It's obviously there's been a lot of failed attempts to treat even people with mild cognitive decline that have Alzheimer's disease. So is this something that you see coming into clinical practice? This is, what are your thoughts about that?
No, that's a great question. Just to add something from the previous question, because people don't know, is we have screened people by the major genetic risk factor that for Alzheimer's disease, which is apolipoprotein E4. So we know that the people carrying this particular gene have more chance to develop Alzheimer's disease than other people. And these people, they have much more vascular problems in the brain. And they are still cognitively normal. So we can detect, as you said, perhaps, I don't want to speculate, but we know roughly that at least we can detect 10 years, if not more, a prior cognitive decline. We can detect those vascular problems. So as you said, there is a window where we can intervene. And if we have the right therapeutic, we can possibly protect the vessel. And what I used to say, seal the barrier to avoid this leakage. So the way I see it, so first of all, the research is ongoing right now. There's several targets that are very promising, currently tested in both animals and clinical trials to try to protect these vessels, because it's not only a question of leakage, but if the vessels are not functional, there's not enough blood flow throughout the whole brain, so which is a big problem. So I see that has two vascular issues. Blood flow is reduced, plus the leakage of the barrier. So we know that we have a few targets that basically target different cell types that comprise the neurovascular unit. So there's the endothelial cells that are forming tubes. Basically, that's where the blood is flowing. But around these vessels, there is multiple layers of different cell types. One of them that is, at least to me, one of the most important one is called the pericyte. So they're wrapped around the blood vessels in the brain. And those cells are very sensitive. They detach the vessels and they die very early in the disease. And if they are not there to maintain the integrity of the barrier, we know that there is these leakages. So the whole idea of different labs in the world is to make sure that these pericytes wrapping the smallest vessels in the brain don't detach. They have to stay there to maintain the integrity and to avoid leakages. So there is some targets, either targeting the endothelial cells themselves or targeting the pericytes directly. There's gene therapy, there's a lot of things ongoing, stem cell therapy and things like that are very promising. So I would say it will happen tomorrow, in the sense that it's going to take a while to do the clinical trials, but I want to stay very optimistic for the people listening, because we are talking about less than 10 years, I would say. If these targets are successful, which they already are in animal models, we're going to have some sort of intervention to fix the blood vessels in the next few years. So I think it's very important. And to do that, the critical part, as you said, there's a lot of failure in anti-amyloid treatments. We have to design the trial very, we have to really think about the design of the trial and what are the category of people you want to treat, right? And I think one way you said is screening people by APOE genotype. So if you have the genetic risk, you could get the treatment. But again, we are talking about 1/4 of the population of the world having the APOE4 genotype. Or 1/3, there's heterozygous and homozygous people. So I don't think it's realistic to just screen the entire population and just give them a treatment to fix their blood vessels. What would be a bit more realistic is to do a combination of biofluid biomarkers that are quite where we can see whether there's things that starts to go wrong in your brain. And when we know that there is something we can do to fix the vessels before there is neurodegeneration and cognitive decline. So I think it's more like maybe screening for ApoE, but also screening for other things around that will tell you, okay, this particular individual will benefit from getting a drug that will fix the blood vessel. And I truly believe, based on the animal experiment and based on the current literature and clinical trials, that if we fix the blood vessels, we may not be able to cure, that's a big word, cure Alzheimer's disease, but at least we will be able to postpone the symptoms by quite a few years, for sure, which is a big deal, obviously, for day-to-day, when people live with dementia on a daily basis, it's a nightmare. So if you can have a decent life for another 10 years, that's a big progress. Hopefully we want to cure the disease, but I think we are going to the right direction.
So a couple of questions kind of stemming from, what is causing the pericytes to detach or causing the endothelial cells to, become, leaky in the blood-brain barrier. If you have someone with ApoE4, you mentioned that's happening sooner. I mean, it's very detectable in people with ApoE4. And also, is the pericytes detaching? Is these signs of, I guess, what you might call leaky blood-brain barrier? Is that something that happens in normal aging, or is it something that happens in abnormal, brain aging that is almost certainly going to lead to cognitive decline? Because the reason I'm asking is, with the amyloid-beta plaques, which is one of the pathological features of Alzheimer's disease, you find them in cognitively normal people. And it's a little perplexing, right?
Yes. No, I think, so to answer that question, so there's a bit of controversies in the field, but most of the literature says that, yes, pericytes they are a particular cell type that are quite vulnerable and quite sensitive. So they do detach and they do die as you age. Of course, we are not talking about that and the extent of what we see in dementia. But we see, just to give you some numbers which will speak to people, we know from mouse studies and human studies, post-mortem brain tissue analysis, we can see that when we look at those pericytes around the microvasculature, there is roughly a 10% to 20% loss of these cells just with normal aging. It's more complicated than that because there is brain regional differences. So you have some regions that lose more than others. Let's say if I talk again about the hippocampus, there are reports showing that there's fewer pericytes in the hippocampus than in the cortex of the human brain at baseline. So that's why I was saying at the beginning, there is different blood-brain barrier properties. There's different cell types, different numbers, probably different functions. So we know that these pericytes, some regions have fewer than others, but they do tend to detach and die with normal aging. The reason why they detach, if I had the answer, I wouldn't do my research right now. We have some answers. The first one is there is a very close crosstalk between the endothelium, so between the blood basically and the pericyte. There is the endothelial cell. So the endothelial cells and the pericyte crosstalk a lot. And I didn't mention it, but pericytes are here to wrap around the vessels and make sure there is no a strict barrier between the blood and the brain, so nothing can cross easily, except glucose and things that we need, oxygen to the brain. The second major property of a pericyte is that they do have contractile proteins, meaning that those cells are able to constrict vessels and dilate vessels. So if they are not here, you can easily assume that your blood flow will be disturbed. So they are here to contract, to constrict and dilate vessels. So obviously when they detach, not only you have the leakage of the barrier, but you also have some flow problems. One of the hypotheses that we are working in the lab, but it's not only an hypothesis, there is kind of growing evidence on that, is the leading cause of pericyte loss might be that the endothelial cells themselves, so they are what we call in a homeostatic state. So they are normal. They express certain proteins, certain transporters that are there to make sure they transport oxygen and glucose, as I said, and other things. As you age, these endothelial cells will turn to pro-inflammatory phenotype, and that's a normal thing. As we age, there's a phenomenon called inflammaging. So it's a contraction of inflammation and aging. And there is several things happening as we age. There's what is called the 7 pillars of aging, which basically there's 7 things that overlap between normal aging and neurodegenerative diseases. And out of these 7, the central one is inflammation. So as we age, we have more inflammation going on, and we know that there is more cell adhesion molecules to site 1. So there is cell adhesion molecules that start to be expressed at the endothelium, which is normally not there, or a very minimal quantity. And we start having significant amount of these cell adhesion molecules throughout the whole body. And the brain is very sensitive to that because these cell adhesion molecules, what they do is they collect the immune cells from the blood, to bring them into the brain. The more you have, the more inflammation, what we call neuroinflammation, the more inflammation you have in your brain. So that's a normal aging process. And the fact that those endothelial cells turn into a pro-inflammatory phenotype, the pericytes that are right next to them so there's a crosstalk there. I won't go into details, but the pericyte will have to detach because to let the immune cells go through. Physically, if the pericyte stays attached to the vessel, there is no possibility for the immune cells to go through and do their job. So the fact that there is more inflammation of the vasculature as we age, there is more detachment, I would say physiological detachment of brain pericytes. That's one thing. And there's some report that as we age, there is less, we don't have the capability, the pericytes don't have the capability to reattach as we can do as we are young. So I don't know if that's clear, but at least there is some hint, that tells us that the fact that the endothelial cells become more inflamed in a way, the vasculature is inflamed, the pericyte will react to that in a bad way. And so they will start shortening their processes also. So I didn't mention it, but pericytes, there's a small cell body with a process, like small processes, like an octopus kind of thing, sitting on top of a vessel. And all these processes are here to push the blood flow, basically, and maintain the integrity. And these processes will shorten because of the inflammation. And of course, if the pericytes are shortening their processes, the flow will reduce. And if it's a chronic thing, if we think about chronically having too much inflammation at the vasculature, the pericyte will chronically detach more and more and shortening their processes. So the leakage of the barrier will come at some point because the tight junctions and everything that makes the barrier intact will start to degrade over time, and the flow will be drastically reduced. So short answer, I'm speaking maybe a bit too much. Inflammation of the blood vessels might be one trigger, and that's something we can target therapeutically relatively easily by injecting drugs IV, right, intravenously. So that would be a perfect way of targeting the vessels to improve vascular function, hopefully.
I think also the fact that you're mentioning the role of inflammation being so critical and key in this process kind of makes me think about lifestyle factors, of course, that aren't necessarily the same as therapeutically targeting with the drug, but omega-3 being one of at the forefront of omega-3 transport has been shown mechanistically, at least in animal studies, to regulate blood-brain barrier function through the MFSD2A transporter. And it's also like many of the metabolites of both DHA and EPA, the marine omega-3 fatty acids, are involved in resolving inflammation. So they have resolvins, they have the specialized pro-mediating molecules, the SPMs and the myrisins, do you think there could ultimately be clinical relevance for omega-3 status in regulating dementia-associated, blood-brain barrier leakiness, in humans, or maybe something worth exploring?
No, I cannot agree more. I think there is more and more studies coming up nowadays, omega-3. And you mentioned MFSD2A, which is one of the markers we are studying carefully because it's specific to the smallest blood vessels in the brain, so the capillaries, and that's where most of the pericytes are. And there is recent studies that shows that as we age and with dementia, MFSD2A, so the receptor for omega-3, it's reduced at the blood vessels. And there is also a link that where there is a reduction of MFSD2A on blood vessels, that's where we see pericyte loss. So we can almost connect what we were saying earlier. Of course, these are just a few studies and it has to be confirmed. But apparently, there's more inflammation of the blood vessels, to go back to the first question to link it, which as we age, also MFSD2A transports and other transporters, not only this one, but this one in particular, is decreased at the capillary bed, which have an impact apparently on pericyte function, because we can see that this hotspot of MFSD2A loss are also hotspots of pericyte loss, which means that where we have the leakiness of the barrier. So yes, I cannot agree more. I think we have to study a bit more DHA omega-3 and how this impact blood-brain barrier functions, because that could be some preventive interventions, things like that, even like targetable drugs, like with drugs. But I cannot agree more. And we can talk a bit more about this, but just to go back at the beginning of your question, you were mentioning exercise. And I think exercise is very important. Even me at mid, I'm not too old. I'm not even 40 years old. And I try to practice every week, knowing all the recent data. So we have to do some exercise, some aerobic exercise, to make sure that the vessels, we have tiny blood vessels in the brain. So if you don't exercise, if you don't make sure that your heart is pumping at a high rate regularly during the week, those tiny vessels that are even smaller than your hair in terms of diameter, they will start to collapse. And you're going to have, and remember, the capillaries, it represents 90% of your brain vasculature, all these tiny blood vessels. So if you don't do enough exercise, you will start chronically to have some vessels that will basically constrict and collapse and disappear. Meaning that the surrounding neurons that are here, and they need oxygen and nutrients and everything from these vessels. If these vessels disappear, you're going to lose neurons, right? So exercise is the number one thing. You have to do that, not only for the brain, obviously, for the heart. It's true for the eyes, obviously. If you don't exercise, you're going to tend to lose eye vision quicker than a normal person as you age. But very important for the brain. You're gonna turn, if you're prone to go to cognitive decline because, let's say, you have a major genetic risk, but if on top of that you don't exercise, you're gonna accelerate, as we know from studies. So I think exercise right now it's highly funded. There's a lot of work going on, and that's one of the biggest thing we have to put that, we have to put to the world, like, okay, need to exercise that no matter what. I mean, if you want to stay healthy in terms of brain function, no other choice.
I could not agree more. When you're talking about if not exercising and how the small vessels lining the brain, they start to get, basically go away. Is that small vessel disease or is that something different?
So we have to be careful. It's not like a person, if you don't exercise, you're going to get small vessel disease. I mean, it's a bit more complex. And I think that's a word I didn't say at the beginning, but this dementia, Alzheimer's disease is one, but the second major form of dementia is cerebral small vessel disease. And I forgot what I wanted to say. What was the question again?
I was asking about what is small vessel disease?
Yeah. Okay. So I wanted to mention, don't want people to freak out. Of course, it's a multifactorial disease. It's not like if you have a genetic risk factor, on top of that, you can kind of counterbalance that by doing more exercise and eating better and stuff like that. But we can discuss about this. But small vessel disease is very different from Alzheimer's disease, although there's a lot of commonalities in terms of brain imaging. If you look at two brains, Alzheimer's and small vessel disease, you're going to see a lot of similarities, meaning that you're going to see what we call microbleeds. So we were talking about blood-brain barrier breakdown. So if there is a significant breakdown of the vessels, you're going to start having red blood cells going to your brain. So that are detectable using MRI. So we can see those microbleeds. That's one feature. They also, small vessel disease compared to Alzheimer's disease, they tend to have small strokes, like what we call lacunes. So they have small spots, small lesions that we can detect in the brain, which chronically and over time you will have a deterioration of your cognitive functions. The third thing that is very important is white matter hyperintensities. So these are white matter disease. It's a hot topic, a hot research area because it overlaps. As we age, we have white matter disease, normal aging. We do have some. Alzheimer's disease, there is quite a lot of white matter. Small vessel disease, there is a lot also. And blood-brain barrier breakdown, obviously. In Alzheimer's and small vessel disease, these are key features of these two diseases, especially to explain the white matter hyperintensities. These white matter diseases are from a vascular source. So it's still ongoing research, but we know that these areas do have leaky vessels, possibly the pericyte detach, where is leakage of blood toxins into the brain, which will damage the myelin sheets and all the axons and the white matter fibers that makes your cognition running properly. So these are the features of small vessel disease. And small vessel disease is also the common cause of stroke. So there's many small vessel disease. We have a cohort here in the UK that we are following with the leader is Joanna Wardlow. So I invite people also if you want to learn more about small vessel disease, she is probably the world expert in small vessel disease. But that's a disease that is independent of amyloid. So that's the big difference, obviously, from Alzheimer's disease. But they do have, the microvessels do have some problems. And I can tell some of them, which we've talked a little bit, the fact that the endothelium gets pro-inflamed. So that's something that is very present in small vessel disease. And to give you an example, I told you about the cell adhesion molecules that are expressed at the liminal side of the microvessels of the brain. When they get shedded, they are released into the blood, right? And we can measure them. And we know that people that do have small vessel disease, they have very high levels of soluble form of these cell adhesion molecules that we can detect in biofluids. So that's already a feature. So what we call endothelial dysfunction. So the endothelium of the blood vessels are getting very inflamed and they don't do a proper job, which imply also, and there is some evidence that the pericytes surrounding them are also dysfunctional very quickly in those diseases. And we can measure one, I don't know if you mentioned that today, but the soluble form of a very complicated name, PDGF receptor beta. So it's platelet-derived growth factor receptor beta. That's a receptor on the pericytes. And that's receptor, same thing. It can be shedded and released into the plasma and into the CSF. And that can be measured. So basically, we know in small vessel disease, they have high level of pericyte dysfunction and high level of pro-inflammation of the vasculature. So and those things, we can also, what we do nowadays is we try to relate those biofluid biomarkers with the neuroimaging features that we see in people to try to correlate whether there is if you have high level of pericyte problems, do you have more white matter disease and things like that? So we try to relate what we see in the blood and CSF to what we see in the brain. And obviously we look at cognition, whether there is some predictive value or whether there is some correlation or association between these biomarkers and cognitive problems. But now we are fairly confident that targeting somehow the blood vessels, the microvasculature in small vessel disease is the way to go. We have to fix these blood vessels very quickly to make sure that these people don't go into small lacunes, microbleeds, and other things that will make them deteriorate very, very quickly in terms of cognition. And the last thing maybe I forgot to mention is this is the sporadic form of small vessel disease, but there is also the genetic form. Same with Alzheimer's. We haven't talked about this, but there is the sporadic form, the most common form, and the genetic form. So in terms of small vessel disease, there is two diseases that are quite known now and quite studied. One is called CADASIL. The other one is called CARASIL. And interestingly, these two diseases involved pericytes and vascular smooth muscle cells. So these are the two cells of the brain that are basically wrapping around the vessels. And it involves aggregates. I mean, for CADASIL, it's NOTCH3 proteins that aggregates in pericytes and vascular smooth muscle cells, which makes vessels dysfunctional very quickly. And those people develop white matter disease at the age of 30, 40 years old, and they go towards cognitive decline very rapidly. It's very severe. And CARASIL, about the same thing. It's HTRA1 protein that is involved. But there is also the genetic form that has exactly the same features: white matter disease, microbleeds, lacunes, and blood-brain barrier issues. And we can measure that. We have all the tools now to measure those things very early before people really go to cognitive problems. So, which means, again, we have to target those vessels to make sure they function properly.
How common is small vessel disease, and how common is late onset small vessel disease, and how do any of those people actually that have small vessel disease end up getting Alzheimer's disease?
So common. It's very common because AD and SVD, so Alzheimer's disease, small vessel disease, are two major forms of dementia. So it's very common, more common than we think. And what you said, the second question was about, sorry?
If people that if you said they're two separate diseases, but I was wondering if people that do develop small vessel disease end up sometimes getting also Alzheimer's, like if there's any...
Okay. So yes, that makes the research more complicated, right? Yes. To give you an example, we have the UK Biobank here, where we have brain tissue samples where we can really look at the features of these different things, Alzheimer's, small vessel disease. And a good proportion of small vessel disease diagnosed patients, they do have some sort of CAA, that's what we've talked about earlier, cerebral amyloid angiopathy, and some of them have amyloid plaques. So that's the complexity of dementia. There is no pure Alzheimer's disease, there is no pure SVD, there's always other neurological comorbidities that are coming into the play, and so it's difficult to disentangle things. But at least when people are alive, there is good imaging tools and biomarkers that we can measure in blood and CSF that tells us who is what. But some people have both, and that's true. And I couldn't say, I'm not an expert on that, I couldn't say the ratio of how many can have both. Some people have SVD and Lewy body. Some people also have some Parkinsonism traits. So unfortunately, it's very common that we can have two forms of dementia kind of simultaneously. But yes, I hope that answers your question.
It does. Circling back to something you mentioned sort of briefly, when you were talking about blood-brain barrier not functioning well, you mentioned glucose metabolism and you decrease, it's important for the blood-brain barrier being intact is also very important for, glucose transport. Reduced brain glucose metabolism can be measured, up to decades even before any Alzheimer's disease symptoms or dementia symptoms occur. Hence, you hear the term type 3 diabetes. What effect does breakdown of the blood-brain barrier have on brain glucose metabolism?
That's another great question, and ongoing studies are tackling this because it's not that clear and quite controversial. Just to tell you a bit more, so the main transporter of glucose at the blood-brain barrier to make sure glucose comes to the brain is GLUT1. So that's a receptor, GLUT1, that has been found to be reduced in Alzheimer's disease. So people have looked at, again, postmortem brain tissue bank and looking at the microvasculature and bigger vessels. And they found that Alzheimer's patients, they have much less GLUT1 at the blood-brain barrier, which means that there is less potential of the flowing glucose in the blood to penetrate the brain. So that's one thing. And we can measure that with several techniques, but one of them, so there's SPECT and PET, so positron emission tomography. And we can measure what we call FDG, which is a marker of what people say neuronal activity, things like that. So we know that when we inject that tracer, it's fluorodeoxyglucose tracer into a patient. If you have Alzheimer's and if you don't, you will have much less FDG signal if you have Alzheimer's compared to a control. But what is interesting, and there's a bit of controversy, I don't want to tell too much about this, but people are explaining this data just saying that there's less neuronal activity, and that's all. But what they don't take into account is these people have less GLUT1, so there is less probability that the radioligand will go. There is less GLUT1 receptor into the brain. So I think the FDG PET signal has to be carefully analyzed in the sense that it could be neuronal activity, but it also could be indirectly the marker of vascular problems because of what I've just said. But again, there is experts in the PET field that will tell you the reason why it is not, and vice versa. It's very technical, and I don't have that expertise. But again, we know that there is less GLUT1 independently of the reduction of the vascular network in Alzheimer's disease. So I think glucose is playing a big role here, and it's currently investigated. I don't know much more than that. Sorry.
Well, I think it's very interesting, and it certainly doesn't seem like we have the consensus or on the answer to that question. Yeah. I'll just mention I remember reading a couple of studies years ago where animal studies where omega-3 deficiency caused a reduction in GLUT1 transporters in the brain. Again, of course, omega-3 deficiency also breaks down blood-brain barrier, so it's kind of what's first, is the glucose, reduced glucose getting into the brain, affecting blood-brain barrier? Is blood-brain barrier affecting the glucose transporters, or both? It seems tricky to sort of figure out, but
that's the chicken and egg question all the time. Same thing with the. I'm not an expert on omega-3, but it's basically the same thing when it comes to pericyte loss, endothelial activation, like pro-inflammation, breakdown of the barrier, loss of blood flow. What is happening first? It's always difficult to address those questions because in human, most of the clinical studies are cross-sectional. You look at one time point. Now, I think there's more and more studies that follow individuals longitudinally with scanning, with looking at biomarkers, looking at neuropsych testings. So we're going to get more answers very, very soon. So there's big centers working to that. But for the omega-3, I'm not surprised that you said that omega-3 deficiency leads to a reduction of GLUT1. Is that what you said? Yeah,
yes,
it has been
correct.
shown. I haven't read that paper, but it goes well with what we said earlier, right? There is a vicious circle where I think it's likely that, of course, the endothelial pericyte crosstalk should be very, very involved in that problems, in my opinion. And that's something I would love to study.
I'll send you my paper. I wrote a sort of interactive review article back in, gosh, it must have been like 2018 or something, I think. And it was on the important role of MFSD2A and omega-3 in ApoE4 carriers. And I have references because for a lot of the studies, like the deficiency in omega-3 causing glucagon transporters to go down, and so I'll send you my review. You might be interested in. Of course, it's a little bit a little bit of a hypothesis sort of review, so I review the literature, but there's a lot of my sort of thoughts for people that are actually exploring the field, because I'm not doing these studies, so you might be interested in that.
And it's just, sorry,
go ahead.
Just rings a bell. I just remember reading a couple of stories, and it's very relevant to what we do in the lab, is that, omega-3, the fact that you give omega-3 to aged animals, and I told you, as you age, you have pro-inflammation, like an hyperactivation of the brain endothelium. And a couple of studies, I've given some omega-3 to the mice, and I remember seeing a reduction of one particular protein that I really like to study, and we are currently studying, is VCAM1, vascular cell adhesion molecule 1. And omega-3 was able to reduce these levels. And we know in the lab, that VCAM1 plays a major role upstream of pericyte detachment. So again, it's there's a lot of things that can be connected. I think it's very interesting. And just another word on VCAM1 is Tony Wiscore in Stanford University, one of the big lab working on proteomics and all these fancy omics techniques. He looked at Alzheimer's and healthy controls. He looked at their plasma, and he has looked at using proteomics, looking at different proteins, which Alzheimer's, and normal aging. And what he found, I think the most striking finding was normal aging. He found, I think, 30 plus proteins in the plasma that were elevated with normal aging, that were related to blood-brain barrier. And if you look down, I think the top 5 candidates were proteins that are part of the endothelial cells, obviously. But the number one that stood out as the number one protein that is elevated with normal aging was soluble VCAM1. So everything, if you put some studies together, it kind of makes sense. There is a bit more to dig, but I think it makes sense, and it might go well with also what omega-3 is doing to the vasculature. So that'd be nice to dig further.
It's very interesting that you mentioned Tony Wiscore as well, because if I recall correctly, he's also been involved in research where he's transplanted young plasma into older mice, and it's sort of rejuvenated in the brain. And it's like, well, are there proteins? I think the opposite was done as well, whereas old plasma was transplanted into younger mice, and it sort of accelerated. Yes. And I think, if I recall correctly, blood-brain barrier breakdown was part of that. So it's like identifying those proteins that it's causing
I think he's doing a fantastic work, and yes, correct. He's looking at the proteins that we have in the young blood that will help not only brain functions or anything like to help not aging too fast, but also we found a few key proteins that are involved in maintaining blood-brain barrier as we age. So I think it's very interesting. And he's using also the parabiosis model where he can link age and young mice. But I think it's very important studies.
Yeah, and the other interesting thing, this kind of brings me back to some of your work where, I don't know if I think you mentioned this very early in our discussion about, blood-brain barrier, when it becomes quote unquote leaky, this allows molecules that usually don't pass into the brain to then pass. And you had a paper where you had published a couple of years ago with, I think it was with your postdoctoral mentor, Dr. Slovovic, about a protein called fibrinogen, which interestingly enough is also like when I go and get my inflammatory biomarkers measured, I like to look at fibrinogen as well as high sensitivity C-reactive protein. It's an inflammatory marker. I mean, it's involved in blood coagulation, but you found this in the brain. What is the significance of that?
Yes. Yes, and it's not only 2 years ago. I think there's, I think the first report, it's probably 10 years ago, but the first report was on, again, brain tissue samples, very valuable tissue samples from donors, where we see that if you compare a control brain, someone cognitively normal, no issues whatsoever, and an Alzheimer's brain or a small vessel disease brain, you start seeing what we call this extravascular deposition. Fibrinogen is one of them. It's one protein that is supposedly in the blood. It shouldn't be in the brain at all, but we start seeing this extravascular deposition of fibrinogen, which means that to cross, you have to have some degree of breakdown of the barrier. So it has been found in Alzheimer's disease. We also found that in animals that either do have Alzheimer's disease or have some sort of blood-brain barrier issues. And we know that this protein that is important for blood coagulation, as you said, and inflammation, has nothing to do in the brain, and it's toxic. It's just toxic. And we found that it's neurotoxic, so toxic to neurons. It's also toxic to oligodendrocytes, where we found that the oligodendrocytes are basically cells that are making up myelin and make sure that the white matter is intact and we can function properly. And those oligos are crucial in the brain. And we show that when you have a leaky barrier and you have fibrinogen going in, the oligos are very sensitive to that fibrinogen. So they take it up, so they internalize fibrinogen, and they die by what we call autophagy. So it's almost like a suicide kind of death, and which leads to white matter disease. And if we go back, white matter disease is a common feature of Alzheimer's, small vessel disease. And as I said earlier, also it's the source of white matter disease is likely a blood-brain barrier breakdown. So fibrinogen might play a role in this formation of white matter disease. So yes, it's a very important protein that we found. Interestingly, in animals, we were able to reduce fibrinogen levels systemically in the blood. Of course, at a level that you don't want to increase to have some coagulation problems or increase bleedings and things like that. So you have to reduce at a level that you don't start doing bleeding or clotting. But just by doing just by reducing the fibrinogen level in a mouse model that do have blood-brain barrier issues, we were able to demonstrate that there is less, obviously, makes sense, less fibrinogen going in to the brain, less white matter damage, and also interestingly, by reducing fibrinogen, we were able to partially restore vascular functions in terms of blood flow and also integrity of the barrier. So fibrinogen has probably different roles, not only coagulation and inflammation, probably a bit more. But I think that's one other way we can target toxin things in the blood to avoid doing damage to the brain. I think it's probably easier to fix the blood vessels rather than doing that. But at least it's another evidence that, okay, a leaky barrier is leading to damage to the brain. And you don't want that, obviously. And the last thing on fibrinogen is, just remember now, fibrinogen can activate the brain-resident immune cells that are microglia cells through CD11B, so that's a specific receptor. So when fibrinogen gets in, it can bind to microglia, so it will induce an overreaction, overinflammation of the brain, which will be detrimental for many things, for the functions of the cells surrounding. And that reminds me that there is a fantastic researcher, Katerina Casudlo. She is at Gladstone University, UCSF area, where I think she developed an antibody that blocks the interaction between fibrinogen and microglia to avoid that overexpression of inflammation or overactivation of microglial cells, because she's also a strong believer that vascular dysfunction are very early and a major contributor to dementia. So that's very interesting research.
It's very interesting. Again, like you said, it really shows the importance of maintaining the blood-brain barrier so that it doesn't allow, things like fibrinogen, which is involved in blood coagulation, to cross into the brain. And again, as you mentioned, the inflammation, it's activating microglia cells, and it's causing that whole process you described earlier, the inflammaging and pericyte detachment, perhaps, and all that. It also, as I was reading and doing some background research on some of your work, and because I knew of the inflammatory role of fibrinogen, I'd already been familiar with that protein, separate of what happens when it gets into the brain. I was looking up omega-3 because I had remembered coming across some studies with it. And interestingly, so air particulate matter. So when you have like air pollution, particulate matter, when people are exposed to high amounts of it, it causes their fibrinogen to go up, right? It's, again, an inflammatory marker as well. But people that were taking in high amounts of omega-3, it blunted the increase in fibrinogen in plasma. And so it would be very interesting to see if in some of the animal models you were discussing, if omega-3 could blunt, the white matter dysfunction caused by fibrinogen getting into the brain. So again, it's another prevention, more, easier, low-hanging fruit thing that people can do now, right?
Yes,
making
exactly.
sure they're taking in enough omega-3.
Yes, exactly. No, I cannot agree more. We don't need to repeat what we said, but yeah, there is this kind of vicious cycle where omega-3 may play a major role into vascular functions, of course. It's not even may play. I think that it does play a role. And it might play even a more important role in people at risk for Alzheimer's. It's possible, like the ApoE4 carriers that we've talked about. And interestingly, you mentioned airborne particles like pollution, right? And I think it's linked to what we've said earlier. So there is Alzheimer's, to keep it simple. If we think about Alzheimer's, it's a multifactorial disease. So you have genetic risk, possibly. You have lifestyle, exercise, and everything. But there is also what we call environmental factors, and pollution is one. And there is multiple studies, as you know, showing that if you live in a big city, you have more chance to develop some sort of dementia. And I'm part of two studies that we've published 4 to 5 years ago, maybe, where we exposed mice to airborne particles from Los Angeles. We're taking from the highway the particles and giving it to the mouse to see what's the impact of the pollution on vascular function. Because there's some evidence also using scans. Those people tend to have more white matter disease and things like that. So we started looking into that, and we found, very interestingly, we found a high peak of vascular. It's a longitudinal disease, but we very rapidly, a few days after giving the particles to the mouse, we were seeing blood-brain barrier breakdown and pericyte degeneration very quickly. One of the first signs that we could see. Of course, now there is debate on the mechanisms, whether the particles can penetrate into the brain through the olfactory area, or whether it's directly the particles in the bloodstream that activate, maybe, as we said, the endothelium a bit too much, which will indirectly cause some vascular issues. That, I think, there is more studies going on. But definitely on environmental factors, and pollution is a big thing to take into consideration when it comes to dementia and especially vascular function, because we breathe that every day, right? So this has to do something with vascular. So it's another factor, but I guess it's not good. I guess if you have ApoE4 and not doing exercise and living in a big city, you probably have much more chance to develop some sort of vascular problems, as we know. But we try to understand how this is happening.
Yes, I remember reading a study where babies, in Mexico City, postmortem, they found, unfortunately in babies, they had like all sorts of amyloid plaques in their brain. And I don't think that's normal. And Mexico City is like,
it's highly polluted.
Highly polluted, yeah.
Yeah,
it makes me wonder about filtering out the air. I mean, if there's some HEPA filter or something that helps a little bit, as well as omega-3 has been shown. I know I keep going back to that, but it's been shown to help blunt some of the air pollution. I know it's the research shows it. So
yeah, I mean, I don't know. I mean, that'd be nice to. I'm pretty sure people are working on that, but there is some countries or cities that are wearing masks and things like that. In China, for example. So that'd be. I guess it's hard. It will be a long study, but I guess we could compare highly polluted cities and people wearing filtering and masks, things like that, to see whether they can preserve some better vascular functions and things like that. But yeah, I guess there's probably ongoing studies that, yeah.
Going back to the ApoE4, because it is such a big genetic risk factor for late onset Alzheimer's disease. And, as you mentioned, at least about a quarter of the population has one allele. I know you had done some research. We talked about how blood-brain barrier seems to break down. At least you're able to measure it, in earlier and cognitively normal individuals. But you also mentioned in one of your papers there was a protein, cyclophilin A matrix metalloproteinase, as a protein that seems to be really involved in, it's an inflammatory protein. But how does it affect the blood-brain barrier?
Yeah. So in the same paper we were mentioning earlier, that people carrying at least one allele of ApoE4, they tend to have more leakage in the medial temporal lobe that we can see with MRI and biomarkers. But of course, it's a great descriptive study, but we went deeper to try to understand how this is happening. And what we found is the people carrying the ApoE4 allele. So ApoE, you have to know that the major source of ApoE are astrocytes. So when astrocytes makes ApoE, so if you have ApoE3, makes ApoE3, if you have 4, makes ApoE4, we know that there is a different affinity to one of the ApoE receptor that is called LRP1 on pericytes. So astrocytes and pericytes are close to each other. They are here to maintain the vasculature as well as neural function and neurovascular coupling. But the ApoE4 is, I don't want to say anything wrong, but there's a different affinity to the LRP1 receptor. You have less chance to bind to LRP1, which will induce a cascade within the pericytes that involve NF-kappa-B, it's a bit complicated, cyclophilin A, which then will lead to expression of MMP9 from pericytes and endothelial cells. So basically, if you have the ApoE4 gene, you will induce much more that cascade than people having the ApoE3, because it doesn't bind to the LRP1 on pericyte, so it doesn't trigger the cascade of expressing cyclophilin A, which will release MMP9. And MMP9 is the matrix metalloproteinase 9 that is basically doing two things that you don't want. Number one is disrupting and the tight junctions between the endothelial cells. So all the endothelial cells are tightly close to each other, and there is tight junctions in between. So this MMP9 will eat up those tight junctions. So if you think about this, you're going to get some gaps, and that's what we call breakdown of the barrier. So you're going to start to see leakage. And the other thing is there is a basement membrane. We haven't talked about this. There is a basement membrane wrapping around the vessels and wrapping around the pericytes. And MMP9 is also eating up this basement membrane. So two things that will basically break down the integrity of the blood-brain barrier. And interestingly, MMP9, it's good to go back a few years back. It's a very nice marker when it comes to blood-brain barrier leakage. We know after stroke, when you have a blood clot in your brain, you have within the next few hours, you have a breakdown of the blood-brain barrier and a big ischemic lesion and things like that. And we know that this breakdown is correlated with high levels of MMP9. There's a perfect correlation. of this breakdown. But very interestingly, when you have an ischemic stroke, you have a second breakdown a few days later. So it's what we call biphasic or biphasic, I don't know, that's my French, biphasic breakdown of the barrier, few hours and few days. And same thing for MMP9, 2 peaks of expression. So it's a very nice marker that goes very well with blood-brain barrier property. So we found this in ApoE4, and both MMP9 and cyclophilin A levels were elevated in cerebrospinal fluid of people carrying the ApoE4 gene. So we were talking about 3 to 4-fold, if I remember correctly, compared to people carrying the ApoE3 gene. So that's a major thing, and we also found that we've used pluripotent stem cells. So we also used human-derived pericytes, so iPSCs-derived pericytes in culture. And when you look at them, these are just cultured of human pericytes, human endothelial cells, and different cell types. And we did that for donors from ApoE4 and donors from ApoE3s. And just at baseline, we were able to also see that the pericytes, just at baseline, looking at that, they produce much more cyclophilin A and MMP9 than the pericytes that do have ApoE3. So these are the evidence that we put. And I think that goes very well with that is we published a follow-up study in mice where we decided to target cyclophilin A, to block cyclophilin A, to see whether if we have mice that do have ApoE4, if we block cyclophilin A, can we restore vascular function? And ultimately, the goal is to restore neuronal function and cognition. So we did that. We used a humanized ApoE4 mice. These are mice that have the ApoE4 gene of a human. So we have also the mice that do have the ApoE3. We follow them as they age. We do MRI on them. We look at their blood-brain barrier function, how the blood flow and everything. And we see exactly what we see in human is they have reduced blood flow in the brain. They have a leakier blood-brain barrier. They have some behavioral problems also in terms of cognition, when what we call novel object recognition, novel object location, those memory issues. So these mice are quite, doing what we were expecting to. And we gave every day for 1 month to the ApoE4 mice, we gave an inhibitor of cyclophilin A that is called Debio 025. So that's a drug that is available currently in clinical trial for hepatitis C. So we gave it to the mouse every day for 1 month, and we checked the same thing. We did MRI, we looked at brain tissue analysis, we did behavior, and as we were expecting, just by doing that, we were able, again, it's not a full recovery, but we were able to partially and significantly restore vascular function. So just by blocking cyclophilin A, we were able to restore the tight junctions, we were able to restore the pericyte coverage of the vasculature, and ultimately those mice had less neuronal damage and less cognitive problems. So it's a good paper that says if you target the vessels, you can have a big impact on neuronal function and cognition. So that's kind of the whole story into 2 papers regarding the ApoE4. Yeah.
Is that drug, is it, a couple questions regarding it. So is it the same as the Alisporavir, I think is what they do?
Yes.
It is.
Yes.
Okay. How safe is that? I mean, is that, something, first of all, if you have an ApoE4 allele, it's like, could you take that prophylactically? Would you have to take it your whole life? Is it, that would be interesting.
Yeah, these are good questions. I don't know if I'm the expert to address these questions, but Alisporavir, is it safe? So first of all, I wouldn't say it's safe. So there had been few clinical trials, and unfortunately some of them have been stopped. And I think it was for design for hepatitis C. It's also currently used, by the way, it has been used a year ago for COVID-19 in France, to kind of for the lung, people having very long, a lot of lung inflammation, things like that, I think. But I haven't checked recently. But I know they stopped the trial for one person that had some cardiac issues after the treatment. So I cannot say it's safe or it's not safe. What I think is we need to design the trial and give it to the right people, I think, rather than giving like this. I think you have to go through criteria before being able to give it. I don't know if targeting cyclophilin A is the best approach. At least we know it partially works in animals, but it might be another possibility might be to target MMP9. So these are close to each other. This has been also studied quite a lot in the context of stroke. But as you know, there's 1,000 compounds that failed in stroke for many reasons. But I don't think anyone has tried targeting MMP9 to look at a more subtle blood-brain barrier leakiness and see whether we can kind of seal the barrier. Yes, I'm not. Yeah, so basically, that's the whole purpose of what we are doing right now. Is we know a few targets that might not be the safest or maybe not the best target, but we have a list of other targets that we are studying, which will hopefully do the same thing. We want to restore pericyte function. We want to restore endothelial function, and make sure the blood-brain barrier is not leaky, pushing the flow as you need to for your brain, and make sure also something we haven't really talked, but the transporters of the blood-brain barrier. If you start having a leaky barrier, you're going to have some transport issues. So it comes back to glucose, oxygen, but also amyloid. If you have a leaky barrier and if you have your vessels that are not functional, there's a few receptors, RAGE, LRP1, there's a few others. If they don't work properly, you won't be able to clear your waste from your brain while you sleep and things like that. So you will have a tendency to accumulate more amyloid oligomers and possibly develop plaques. So everything is interconnected. But again, yeah, I think fixing the blood vessels, whether cyclophilin A is the right target, I don't know. But I think it's promising. We just have to see who will benefit from getting the drug.
So if you're having a dysfunctional blood-brain barrier, if you're basically having blood-brain barrier breakdown, everything you've been describing, the glymphatic system, which is when you sleep, it squirts the cerebral spinal fluid into the brain to clear out the amyloid plaques. Is that also impaired somewhat?
So there's active research in this area, obviously, but that's fair to say. Yes, of course, if you have, because at the arterial level, you have the perivascular space, which is enlarged when you sleep. It has been shown. And yeah, of course, if you have disruption of these vessels, obviously the perivascular space will be the properties and the cells being around perivascular macrophages and everything will be highly disturbed. But yes, it's fair to say. It's just the glymphatic system. It's very controversial nowadays, as you may know. We don't really know whether, the fact that we clear things from the arterial level and then it gets, there's a convective flow that drains the interstitial fluid back to the veins. That's kind of the theory of the system, but I think it requires a bit more work to prove that this is truly happening, in my opinion. But there's also other system. There's the IPAD, so that's the intramural periarterial drainage. So there's basically either the lymphatic and or the IPAD system to clear out toxins out of the brain. But again, I'm not the expert on that, but it's fair to say that, of course, if you have vascular dysfunction, the clearance of toxins, not only amyloid, but alpha-synuclein or other things, will be highly disturbed, yes. So,
in my opinion, that sort of puts, of course, the vascular function and blood-brain barrier integrity upstream of amyloid in a way, because you'll lead, having that dysfunction first will then lead to more, amyloid accumulation, if you're not able to clear it out, right? So what do you think of, a there's been a lot of failed attempts to target amyloid beta plaques, soluble amyloid. Although recently there's the lecanemab, which targets the protofibrils, so it's still like a soluble form of amyloid, but there's some positive results with that drug. Do you have any thoughts about it? Do you think?
Yeah, I think I like to be optimistic and I like to be positive. So yeah, I think it's good that we have drugs for not only for research, for people also, for the families affected. It's good that we have a drug that can remove to some extent the plaques, and there's evidence of that. What is a bit less, a bit more controversial is the impact on cognition. So there is some improvement, but it's not what you would hope for. So which goes back to, targeting amyloid only is probably not the way to go. And it's probably not sufficient, or might be too late also. So it goes back to our important research. If you tackle things and target things much more earlier than that, you will have a better chance. So I don't have much to say. I think I would see more like as a cocktail of treatment. If you give something that will make your blood vessels working as they should work in terms of pericyte function, clearance, endothelial function, plus the drug of like an anti-amyloid any form, I would bet that you would have more chance to have a bigger impact, a bigger positive impact, very positive impact on cognition. But again, it has to be done very early. If your brain is full of plaques, it's probably too late already. So that's the importance of the research we do right now. But I see that as a cocktail, maybe improving the blood vessels, because we need those blood vessels to be functional to clear out amyloid. So the combination would be probably a viable solution in the near future, maybe.
So on the opposite end of treatment would be prevention. And I think looking at dementia and Alzheimer's disease more as a vascular disease sort of opens up the doors for a lot of preventative measures. Blood pressure. So it's actually very common, in at least in the United States, almost 50% of the US adults have hypertension, and 20% of young adults do, age 18 to 39. So how does high blood pressure affect the brain and dementia risk?
No, that's very important. So there's I would say the three major, at least in my research, I'm not saying major, are the three major cardiovascular risk factors. Hypertension is one, but there's diabetes and hyperlipidemia. But in terms of hypertension, and I think you've sent me a few papers, but I think it's very interesting to know that if you have early hypertension, so if you're young and you have hypertension, it's possibly more harmful than if you have it later in life. And I guess it's just a question of duration. If you have hypertension sustained over many years, obviously it can impact the stiffness of your blood vessels. It will impact, as we talked, the pericyte function at some point. Bottom line is, yes, hypertension is known to impact the brain, and there's like the basal ganglia and those deep structures that suffer quite a lot from hypertension, where we see also there's always an association. When people have hypertension, they tend to have more microbleedings also in this area of basal ganglia. So yes, it does impact the brain, and we know there is a few studies that shows that hypertension also triggers a blood-brain barrier leakage and pericyte loss. It's not the whole story, because in terms of small vessel disease, if we come back earlier, we have thought that one of the cause, or maybe the major cause of small vessel disease, was hypertension. But nowadays we know that's not the case, because, to give you an example here, maybe a third, even more, of the cohort of small vessel disease patients that we follow, they don't have any hypertension, first of all. So it tells you that there's a big chunk of people that can move to small vessel disease, So meaning having white matter disease that were thought to be due to hypertension and how it damaged vessels. But it's not the case. So that's a bit more complicated. But obviously, it's not good to have hypertension, as we know, for the brain. And there is some studies showing that giving antihypertensive drugs has a positive impact on cognition as well. So it tells you that no matter what, if you have hypertension, you have to try to reduce it, obviously, pharmacologically, because it will have an impact. But interestingly, to just go back at the beginning of my answer, it's a bit controversial, but if you have early, like 30s, 40s, if you have early hypertension, you will have a likelihood and more chance to develop cognitive decline quickly, as compared to a normotensive person of the same age. And interestingly, they were showing also in the paper that if you have hypertension that's during your 70s, if I remember correctly, it doesn't impact your likelihood to develop dementia. So it goes back to, I think just the final point is, I think it's very important, the duration. It's the chronic part of hypertension is very critical. If you have that for many years, same thing for inflammaging. As you age, you have a chronic, slow, low-grade inflammation that will disrupt your vessels very slowly. Probably the same thing with hypertension. If you have too much hypertension, your arterial stiffness will be, disturbed and damaged over time, and this will degrade your blood-brain barrier much faster than a normotensive person. And this has the regional specificity and different sensitivity in the brain, but that's an active area again. We have tools now to measure these brain regions, how they leak, and try to compare to with people with hypertension. But the difficult part in clinical research is no one is hypertension and nothing else, right? Hypertensive and nothing else. There's always comorbidities, obesity, other things. So it's hard to disentangle what pure hypertension is doing to the blood vessels, because there's always other things. You can try to correct statistically with covariates and things, but it's quite hard to study.
So it does seem like, as you mentioned, cumulative exposure seems to be the more important thing here. And again, people that are probably in their 20s, 30s, 40s aren't even, if they have high blood pressure, aren't probably thinking it's a big deal. When in fact, that's probably the biggest deal because they're starting early with the high blood pressure.
That's my thought. That's what we read. Yeah. Same thing with exercise. I think what we hear from these recent studies is the critical window is, I don't know if I'm correct, you may know better than me, but it's like 40, 55, something like that, where you really have to exercise and to come back to the small vessels. You have to open them up and make sure they are functional. That's the critical window. Is that, I think it's partly true for sure, but I don't know, you may know a bit more than me on that question. But yes, I think that's the time that
certainly it's never too late to start exercising, and there have been studies showing even in the 70s you can have cardiovascular improvements. Of course,
in fact,
earlier is better, and exercise, as you mentioned, it is one of the best ways to improve vascular function. It improves blood pressure, arterial function, all those things. And interestingly, the sauna also does that. The heat stress, a lot of the physiological aspects of elevating your core body temperature.
I didn't know that, but that's interesting.
And there's a lot of research. In fact, there's, of course, with observational data, you never can establish causation, as you mentioned. There's all sorts of covariates and things, but there are studies showing that dose dependently, sauna use is associated with like a 60% lower dementia risk and Alzheimer's disease risk if you're using the sauna 4 to 7 times a week. If you use it 2 to 3 times a week, there's like a 20% lower risk after adjusting for other factors as well. But we do know sauna, like there's intervention studies. I mean, it's comparable to moderate aerobic exercise in terms of the effects on blood pressure, heart rate variability and stuff. So also sort of just interesting, but exercise being a really important one for, prevention, dementia and Alzheimer's disease prevention as much as you can do.
Yes.
How does alcohol affect the blood-brain barrier?
Again, I think it's a bit controversial. at least what I know from the literature is chronic consumption obviously is not good. And interestingly, we can see, I don't know if you've seen that, but there is a gender effect, a sex effect. So the males tend to have, if you're a chronic drinker of alcohol, you will have some vascular issues. It has been linked. So if you chronically drink alcohol, you have a leakier blood-brain barrier. It has been shown. But if a woman drinks the same, that's age mass and everything, they will have almost preserved vascular function. So that's very interesting. But it's again a few studies, but the hypothesis that they bring are estrogen and all these that have anti-inflammatory properties and things like that. But so obviously, drink too much does impact your vascular function. So what is a bit more controversial is like moderate drinking. So there's you can see things showing that moderate drinking is impacting your brain function, but not only brain function, but vascular properties of the brain. And there's also other studies that show that moderate drinking doesn't do anything. It's even positive in some cases. So I think I'm not an expert on that subject, but I guess if you go in details into the design of the studies, you may find the reason why these are different results. But that's the results they put up front. So yeah, you probably know better than me what it does, but yes, to some extent, just the alcohol itself impact blood-brain barrier functions just by increasing the level of cytokines in the blood, and which will increase the inflammation of the blood vessels. So yes, and if we go back to dementia and inflammaging, if you do that chronically, obviously, if you have sustained level of inflammation of your blood vessels, we know what it does. It's going to impair pericyte functions, and over time, and possibly leading to leakage, which it's a bit bad. So chronic drinking is of course, people will have a more chance to develop dementia very quickly. So, but yeah, I don't know if you know more than me, but that's what I know about.
Yeah, I know that the same is very there's a lot of conflicting data, and you'll find, again, as you said, some studies showing moderate alcohol consumption protecting against dementia, and others showing it not. As I sort of tried to dig deeper, what I was finding was it was protective in people that were not ApoE4 positive more.
Okay.
And so, and then of course it all comes down to each study, how they defined moderate or light drinking actually was different as well. So
it's not controlled,
it's not, in some cases it's like, okay, well, light drinking is less than 1 drink a day. Or in some cases it was like they studied it weekly. So it's like, okay, less than 3 drinks a week. Well, that means some days you're not having any. So there's so many variables here that it seems challenging to...
But that's an interesting area, because, I mean, I'm just thinking when we tell me drinking, I think about red wine. I'm French. And red wine are full of flavonoids that from the grapes, which has been shown to be very important for brain functions and also for vascular function. So there's also the positive of thinking that way. If you take a little bit of wine, I don't know how frequent, because I don't know the studies and how much you should drink, but at least 1 glass every now and then should be helpful and good for your brain. That's another thing.
So I think avoid daily drinking sounds...
Yeah, of course.
Do you know about, you mentioned obesity and type 2 diabetes and these things also. I mean, does obesity accelerate the blood-brain barrier breakdown or leakiness?
So I've seen a few studies showing that, but also interestingly, I don't know if you know, but William Banks, one of the, if not the one, the most highly cited researcher when it comes to blood-brain barrier. I think he's in California. He also wrote a recent review that blood-brain barrier leakage will trigger obesity and not the other way around. as we age. So it's very interesting. I haven't read the review in details, but yes, I found studies that show that obesity, of course, these people tend to have higher levels of in pro-inflammatory markers, cytokines, chemokines in the blood, and things like that, which will have an impact on barrier function. We know that. But the other way around, I didn't know. I've just found that recently with William Banks. So interesting, but I think he's claiming that there is some like hypothalamus area that are basically, controlling starvation and the way you should eat and things like that. If the vasculature in this area are kind of leaky, that's where you're going to start having some dysfunction in this starvation and food intake and things like that. So people will start to eat too much and things like that. So I think it's interesting. I haven't read the review, but I can send it to you. I think it's an interesting idea. But I think there's some data supporting that.
Sounds like a vicious cycle. I mean, I thought the same thing as you. It's like obesity, visceral fat. I mean, that's inflammatory, environment. So it makes, to me, it would make sense that it would affect the blood-brain barrier in a negative way. What about homocysteine? High homocysteine it's a cause of one of many causes of high blood pressure.
That I don't know much. I'll be honest.
I was just curious, because I did see, like it's also associated with brain dysfunction and dementia, and there was like at least one study I saw where.
I've seen that it's a classic. I mean, not classic. It's a marker that is often looked at, but I'm not very knowledgeable on the pathways that are involved with homocysteine. So
you don't study that.
if you have some, happy to read more about this.
There's a paper I linked you to and some of the notes I sent that people that had high homocysteine, when they were treated with a B6, B12 folate sort of concoction to help lower their homocysteine, it improved their cognition and also markers of blood-brain barrier. So interesting. But I mean, I know it's not the focus of your research.
Good.
Just, I mean, I think this is really great, Axel. Like there has been a lot of paradigm shifts in people's thoughts on Alzheimer's disease, with them thinking about it being a metabolic disease in a way, right? Type 3 diabetes, you'll hear. But like, I think this vascular dysfunction it's so crucial, and may go hand in hand with the metabolism, maybe at the root of it. But identifying some of these early biomarkers that you were mentioning, even in plasma, I don't know how early or if they're even available to people to measure. Are they?
It's still at the research phase. So there's I don't know if you know, but there's a new technique called electrochemoluminescence, and there's two major platforms worldwide. One is called Mesoscale Discovery, MSD. The other one is the CIMOA platform. And the beauty of these two technologies is you can use a small amount of fluid and you can get a lot of data, basically, to make it simple. And so these particular fairly new vascular biomarkers are currently being, validated and everything in those platforms that will be high throughput and where you can imagine to do a lot of patients at a time. Right now, it's quite the workflow. Right now is quite long and difficult. So that's why we try, as any biomarkers, we want, like same as we heard from Japan, I think. You can get, same as you get your blood glucose right from the finger, you can get your amyloid status, right? Amyloid 42 levels now, and things like that, and get diagnosed with if you have Alzheimer's or not from just a drop of blood. So that would be the idea, basically, having this, but this will require probably a few years, obviously. But it's available at the research level, but not at the clinical level as of yet.
What about the soluble platelet-derived growth factor receptor one you mentioned?
So it's ongoing in clinical trials in the US. So I guess it has been done in hundreds of patients, participants, that now they try to go to thousands. So, you need some validation step with a bigger cohort and make sure that this is truly elevated, let's say with ApoE4, with aging, in a much larger cohort of patients. But yeah, I think it's much closer to be available. Hopefully, talking about months, a year or two. Hopefully, yeah. But I'm not
Wow, that's
part
fantastic.
of that validation step. But hopefully, this will come quick, because I think it's a very, very, very valuable tool for us. Yeah.
I'm very thankful for the research that you do in your lab, Axel, and others. Dr. Slovovic and some of the people you mentioned. I mean, just that people are out there trying to identify new biomarkers of dementia and Alzheimer's, vascular biomarkers, and new treatments, and also opening up more preventative measures and understanding it in a different way. So, thank you so much for all the research you do, for coming on the podcast. I know you're on Twitter. I follow you on Twitter. Your Twitter handle is at AXL and then _ Montagne for anyone that wants to follow Axel on Twitter. He tweets interesting studies related to the stuff we talked about today. You also have a lab website, which I was perusing,
Yes.
a very nice one, and montagnelab.com. So that's montagnelab.com. And we'll link to those as well on our episode page.
You.
But really, I learned a lot today, Axel. I really appreciate you taking time to educate everyone on this important role that vascular function plays in cognitive function, in preventing neurodegenerative diseases, and just healthy brain aging in general. So
thank you very much for the invitation. I really enjoyed it. So I hope the listeners will do.